Welcome to Department of Botany, Kalna College. Myself, Dr. Somnath Mondal. Today's topic is Introduction of Plant Biotechnology Part 3. In Part 2, we have learned about the natural plant genetic engineer Agrobacterium tumefaciens and the configuration of TI plasmid and how is it integrated into the plant genome. So today in part 3 we'll study about the Roundup ready soybean. This was one of the first ever transgenic crops, that is the herbicide resistant plant. So what is Roundup? Roundup is a broad spectrum herbicide. Its active ingredient is glyphosate. Glyphosate is an organophosphate compound which inhibits the Shikimate pathway and thus blocks the biosynthesis of aromatic amino acids. Therefore, Shikimate herbicide, if sprayed on plants, glyphosate will inhibit the Shikimate pathway in those plants. Now let's have a look at the Shikimate pathway. Now phosphoenol pyruvate and erythrose 4-phosphate to combine to form 3-deoxy-D-arabino heptulosonate 7-phosphate. Next, it will be converted into dihydroquinonate, which will be converted into 3-dehydroshikimate, followed by Shikimate and Shikimate 3-phosphate. Now this Shikimate 3-phosphate will be converted into the 5-enol pyruvyl Shikimate 3-phosphate and finally into corismate. So we have a very concise account of the Shikimate pathway. So this is not our main concern. So we will look at the problem lies. So in every step of this biosynthesis there will be specific enzymes assigned for. Among them the enzyme 5-enol pyruvyl Shikimate 3-phosphate synthase, or EPSPS in short, is inhibited by glyphosate, that is the active ingredient of Roundup herbicide. Therefore, corismate will not be biosynthesized in plants where Roundup has been sprayed, and thereby aromatic amino acids like phenylalanine, tyrosine, tryptophan will be blocked. Now glyphosate therefore will inhibit the enzyme EPSPS and the corismate could not be biosynthesized. That is the precursor of amino acids like phenylalanine, tryptophan and tyrosine. That is why within hours of Roundup herbicide spray, weeds will stop grow and yellowing will start in couple of days and finally they will die. Now the question is, weeds are affected by glyphosate, why not the crop plants? Because we have done transgenic crops which are herbicide or Roundup resistant. Now let's have a look at the enzyme EPSPS that is blocked by the glyphosate. This enzyme is a 46 kilodalton protein with 2 domains, and 2 domains are hinged at a point. There are 2 classes of EPSPS, class 1 and class 2. Class 1 is found in plants as well as in some bacteria, and this class 1 EPSPS is susceptible to micromolar concentration of glyphosate. However, class 2, which is found only in some bacteria, is resistant to glyphosate inhibition. Hence, if class 2 EPSPS gene can be incorporated into economically important plants and expressed, then they will be resistant to glyphosate herbicide damage, and the weeds which are non-transgenic will be omitted from the field. Therefore, the vector construct for this program looks like there will be the left border and right border tDNA sequences. Within the left border and right border sequences, here marked with yellow lines, there will be CAM35 promoter, that is the universal promoter, that is the constitutive promoter, under which our gene of interest will be incorporated. First of all, there is Petunia chloroplast localization signal. I will come to that. Next, there is EPSPS class 2 gene, and finally there is termination signal. And this vector construct is being transformed into agrobacterium cell. Now, this transformed agrobacterium cell will be co-cultivated, and within plant where the TI plasmid has been successfully integrated, in such plant cells, what will happen? Let's have a look. We have learned about the tDNA transfer and tDNA integration, the whole process, the mechanism behind the function of Vir operons and the non-homologous end joinings in our early presentation. So here we will concentrate only on the mechanism of EPSPS class 2 and how it bypasses the effect of glyphosate herbicide. So, after a successful plant transformation, a plant cell nucleus having the gene construct of with our gene of interest, that is EPSPS class 2 gene, transcription will start and it will form an mRNA molecule with the chloroplast localization signal as well as the EPSPS class 2 gene. Now this mRNA will be translated and the protein, the resulted product, will be EPSPS class 2 tagged with chloroplast localization signal. Now, due to the presence of this chloroplast localization signal, it will give the get pass to the EPSPS class 2 enzyme to go into the plant cell chloroplast. Now, why chloroplast? Within chloroplast, the chorismate biosynthesis pathway is taking place, which is going to be blocked by glyphosate. So, in this slide, we can see there is already the class 1 EPSPS, that is the wild type EPSPS, already present within our economically important plant cell, and the class 2 EPSPS that has been introduced into the plant. Now glyphosate will positively inhibit the activity of class 1 EPSPS, but it will not inhibit the class 2 EPSPS. Thus, even when class 1 EPSPS is unable to form chorismate, but this block will be bypassed in presence of class 2 EPSPS and chorismate will be successfully biosynthesized. Therefore, in transgenic crops with additional class 2 EPSPS, cyclamic acid pathway will carry out normally and biosynthesis of aromatic amino acids will not stop. Thereby, the plants which has been transgenic like Roundup ready soybean, they will not have any adverse effect of the spraying of Roundup herbicide. Only the weeds of that field will die as they have no class 2 EPSPS.